Hello, in this video we will discuss about the antigen representing cell, the dendritic cell. So antigen representing cell is a lot of type which that is present in the different tissue and different site. For function same, for pathogen recognition. So the antigen presenting cell is the innate immune cell present in lung, liver, upper circulatory system and skin. Antigen presenting cell used for the activation of immune response, and the immune response can be innate or adaptive also, because the macrophage and neutrophil also stimulate through antigen representing cell and get the antigen to show the adaptive T and B cell also for the activation to produce antibody, and this antibody will opsonize the pathogen antigen because the against the antigen antibodies. So let's begin to understand the whole story. What is the dendritic cell function and how to regulate, and the T regulatory cell function also. So here you can see the virus and bacteria which that will entering into the blood circulatory system, and this is the harmful for us to cause infection in our tissue, to cause different disease. So for example, here is the antigen representing cell. Dendritic cell will endocytose the antigen, the pathogen, and after this it can be also enter into the because it is a parasite, the antigen, and it will be enter into the tissue to cause disease. And this is the blood circulatory system, and this is the lymphatic system, contain lymph node as well as thymus gland. So the thymoma mean the cancer of the thymus gland, which that produce the T regulatory cell dysfunction. So in this way, here you can see the MSC class 1 molecule will show through antigen representing cell, dendritic cell, and co-stimulatory, and the T cell receptor will naive T cell will proliferate into the T helper 1, T helper cell, and T helper 17 and 1, and CD4 and CD8 and B cell. So the CD4 is the helper and the CD8 is the cytotoxic, and this produce the interferon gamma, interleukin 17 and interleukin 22, which that recruit the other cell, immune cell, while the CD4 will recruit the B cell through production of interleukin 4, interleukin 6, and after this here is recruitment also the CD8 through these cytokines. And this is the thymoma formation will lead to autoreactive T cell production, will lead to autoreact to our own tissue to cause the infection, to cause the disease, is known as the autoimmune disorder. So here is the infected cell tissue, which that will show the antigen through a antigen representing molecular receptor is known as the MSC1. And in this way the MSC1, which they recognize through a CD4 to produce the interleukin 4, 6 anti-tumor necrotic factor for recruitment of the CD8 as well as recruitment of the B cell. And in this way the recruitment of the B cell will produce the B memory cell and the plasma cell. Plasma cell will immediately produce the antibody. So here is the CD4 will also activate the CD8. So in this way here you can see this is the neuromuscular junction which that is present, and the neurotransmitter acetylcholine, when our muscle will trigger to contract with our own choice or the involuntary or voluntary, so it can be bind the neurotransmitter acetylcholine receptor with a muscle cell. So after the binding, the depolarization will occur, but in this case the dendritic cell will recognize this receptor to activate this is the myasthenia gravis problem, that is the best example, and the dendritic cell will recognize and to activate the CD4 cell, and CD4 cell will activate the CD8 as well as the B memory cell and plasma cell through a B naive B cell activation. And the plasma cell produce the antibody, and this antibody will target to the neurotransmitter receptor to collapse, and in this way the dysfunction of the neuromuscular junction will lead to difficulty to breathe, difficulty to muscle movement, and difficulty to speak because the motor disorder. And in this way here is you can see the antigen presenting cell which that is recognized through to the T naive T cell through T cell receptor and MSC class 1 molecule and the co-stimulated molecule. And in this way the basically it will proliferate the T cell into the different cell which that is we had discussed before. So in this way the T helper cell for example produce, and this T helper cell and T regulatory cell proliferation, and this T regulatory cell proliferation which that is will inhibition of auto-antigen reaction. And how it's possible, let's begin to understand. So the T regulatory cell will basically show the antigen to the CD4 to educate, hey, don't touch to our own antigen, our self antigen. So in this way, the CD4 will learn and don't touch to the our own antigen, and the programmed CD4 cell non-auto antigen reactive. So in this way, the T regulatory cell also check the antigen as a neurotransmitter and will lead to T regulatory cell to show the to the CD4 to educate, and he tell them don't react to our own antigen because our own antigen is not a antigen. So here is the antigen representing cell activation, how it is possible. So let's begin to understand. For example, there is a antigen representing cell upper side which engulfs the antigen, and after the piece of antigen will show to the T cell, naive T cell, through a T cell receptor via a major histocompatibility complex are present in the antigen presenting cell. And in this way, the T cell receptor contain alpha and beta subunit as well as the CD4 positive molecule. And in this way, both action will lead to cognate antigen recognition. After the recognition, it will proliferate into the T regulatory cell, to regulate the antigen checking. And after the checking, self antigen checking to educate that CD4 to don't activate the B cell for this antigen because it is not good thing. So here is the CD8, CD80 and CD86 molecule which that is bound with the CTLA4 and CD28. So the CTLA4 is basically co-stimulation will lead to co-stimulate, and the CD28 and that PD1 which that will lead to co-stimulation will lead to interleukin 2 transcription factor will produce after the phosphorylation will bind with the DNA to transcribe and translate the interleukin 2, and this interleukin 2 will bind to other cells and recruitment of the other B cell as well as the CD4 and other macrophage for the innate immune response. So here is the T cell receptor, interleukin 12 and interleukin 2 receptor, which that is basically is the bind with the interleukin 2 via alpha, beta and gamma subunit to recruit the JAK 1 and 3 will docking the stat 3 and 4 5, and after this will bind with the DNA to proliferate cell proliferation differentiation into T helper cell target tissue antigen programmed protein recognition, and in this way the proliferation to form a T regulatory cell, and in this way the basically the antigen representing cell dendritic cell function.